You may find yourself near a fallow field that is a sea of small purple flowers. There's hardly any insects, and it's hard to find any native vegetation once you enter the field of silver-green foliage. While it's been mowed, burned, tilled, and sprayed with herbicide, the onslaught of invasion doesn't stop. Today we are talking about the invasive perennial Russian knapweed. My name is Christy Gladham, and I have worked in biological control for over 10 years. Welcome to the Colorado Department of Agriculture's Biological Control Series, in collaboration with the USDA Western Sustainable Agricultural Research and Education Program. Russian knapweed is the most widely distributed of the invasive knapweeds in the western US. However, it is commonly mistaken. So let's discuss how to identify this weed. Biological controls are host-specific, so Russian knapweed agents will not establish another knapweed infestations. Russian knapweed may be distinguished most easily from other knapweed species by both roots and flower heads. Russian knapweed has black, scaly, spreading roots, while other invasive knapweeds will have a singular large taproot that is creamy white. You can also see a distinct difference in the bracts of the flower heads. Russian knapweed has peepery bracts, while other knapweeds will have comb-like bracts with lateral spines. If these are hard to see in the field, a jeweler's loupe could be used, or you might open a photo app on a cell phone and zoom in. Introduced to North America in the late 1800s, Russian knapweed, Rhoponticum repens, is a noxious invasive perennial that forms large rhizomatous colonies. This means that there's an extensive creeping root system that feeds a lot of clonal shoots above ground. The root system overwinters year to year. The above ground foliage will die back in winter, and new shoots will come up the following spring, typically around April in Colorado. The plants are 1.5 to 3 feet tall with silver gray foliage and small leaves directly attached to the stems. The individual shoots will begin to bolt in May to June and will flower continuously into August. The flowers are typically pink to purple, urn-shaped, and a 1/2 inch to 1 inch wide, or about the size of a nickel. Each shoot has been reported to produce 50 to 500 seeds per season. These seeds can be viable in the soil for up to 75 years and are the likely means of long-range dispersal of this plant through contaminated hay or seed products and movement by farm machinery or vehicles. However, the seeds are not very viable. Most never germinate. The primary means of local spread of this invasive perennial is through its root system. These root systems can reach over 20 feet below the soil surface and spread up to 185 feet in diameter. Root fragments as small as 1 inch can create new colonies, and a new plant has been reported to spread over 12 square meters in under 2 years. The plants discourage foraging animals as they are quite bitter, even toxic to horses. The roots are thought to excrete chemicals that prevent other plants from germinating and growing, giving Russian knapweed an even stronger competitive advantage in the field, reducing native vegetation and devaluing land. Individual colonies can live for 75 years or more. While a singular patch is usually one large individual that sends up genetically identical gunnets or shoots from the root system, new or other discrete patches are often the result of seeding. To prevent spread of Russian knapweed in a landscape, you'll want to control the plant's flowering and seeding. Control and management of a single patch will need to focus on decreasing the carbohydrate reserves in that large perennial root system. As with most perennial colony-forming weeds, it is most important to work on eradication of small satellite patches while controlling the flowering, seeding, and dispersal from larger patches. Once smaller outlier patches have been eradicated, control of large, well-established patches should focus on the perimeter of the patch to prevent spread and begin to reduce the size from the outside working towards the center. Cultural control to prevent, monitor, and quickly eradicate new infestations is usually the best bet with Russian knapweed. For established populations, combinations of treatment over multiple years is usually required. Mowing can suppress growth, but will not reduce Russian knapweed populations. Tillage is not recommended as the fragmentation of the root system will encourage more growth. Hand pulling could be effective on very small and new infestations, but is generally not feasible with established populations. Mechanical disturbance also creates soil disturbance that can encourage more invasive weed species. Burning is not at all effective and can contribute to higher knapweed dominance within a landscape. Herbicide is generally effective when utilized properly, but is not cost effective on already degraded land, and large-scale applications can be quite detrimental to the environment. If you have large, extensive patches of knapweed, and the goal is to reduce the density of knapweed on a longer time scale of say 5 years, have minimal input in either cost or time, reduce invasion potential for neighboring properties, and reduce disturbance of the land as much as possible, then biological control will be a great tool to utilize. As always, before considering collecting biological control agents, please ensure you have the proper permission or permitting to do so, and ensure the biological controls are permitted and allowed in your area. Movement of biological controls across state lines is permitted by the USDA, APHIS PPQ, and subject to rules and regulations. The 2 biological controls I will discuss today are Jaapiella ivannikovi and Aulacidea acroptilonica. Jaapiella ivannikovi, a Rosette gall-forming midge, was approved for release in the US in 2009. These are very tiny flying insects, specifically a type of fly called a midge. Midges are a bit less than 2 millimeters in length or a little smaller than a sesame seed. The adults begin emerging in mid to late spring, but they are very short-lived. Males live slightly less than a day, while females can live for 2 to 3 days. The eggs of the midge are preferentially laid in actively growing shoot tips of Russian knapweed. As the eggs hatch and the larvae begin feeding on the plant, an interaction between the biology of the midge and the plant occurs that forms a rosette gall. These galls are a mass of malformed leaves tightly fused in a rosette pattern. They appear silver due to the hairs and webbing produced by the plant. If you cut open one of these galls, you will find several small larvae that are technically maggots since they are a fly species. The larvae will pupate, turning into adults, and emerge to mate and lay more eggs. There may be up to 4 generations per year, with each generation lasting approximately 1 month. In fall, the developing larvae will turn into pupa, but will stay in a pupal stage over winter. Once temperatures warm up in spring, they will complete pupation and emerge as new adults. Since these midges have such short lifespans as adults and are very delicate, it is best to collect the formed rosette galls before new adults emerge and place the galls directly in the intended Russian knapweed patch for release. The stems containing galls can be clipped and placed in a cup with water like a bouquet. These cups can then be placed around a Russian knapweed patch, hopefully in shadier sites, to let the midges emerge from the galls naturally. This can occur anytime between May and August. The best sites to find rosette galls are usually along riparian corridors and areas where Russian knapweed is regularly mowed. However, when whole galls are moved, parasitoids that exploit the life cycle of the midge could also be moved unintentionally. These parasitoids can impact midge populations, but also native galling insects. It is best to only move rosette galls if you are keeping them in the same general area. They are not the most effective biological controls. While they can reduce flowering and seed set, the proportion of galled stem tips is usually not high enough to make much difference. However, they can be very prevalent in certain situations and can have additive effects when combined with another biological control agent, the Stem Galling Wasp. Aulacidea acroptilonica was approved for release in the US in 2009. These wasps have no stingers and are very tiny. They are also a little smaller than the size of a sesame seed. The adults can be difficult to spot in the field because of their small size. They will be found only in close association with Russian knapweed and will typically be the only active insects in early spring on these plants. The adults emerge very early in spring from galls or malformed plant tissue in the previous year's dried out stems. The galls look a bit like a pea pod contained within the stem, and you will be able to observe emergence holes in the galls where the wasps have chewed their way out. The adults are only active for a few short weeks. Each individual adult wasp only lives an average of 5 days after emerging. They mate and lay their eggs in the new Russian knapweed shoots. The shoots are usually between 4 and 8 inches tall and still quite pliable when the females are laying their eggs. When the eggs hatch, the larva will begin feeding in the shoot. As the plant tries to protect itself from the larval feeding, it forms a gall, or a mass of tissue to contain the larva. New gall formation can be seen in as little as 2 weeks after exposure to the ovipositing or egg-laying females. The larva will feed and grow inside the gall over the summer. The larvae overwinter inside the gall and will pupate to transform into adults in the following spring. The wasps only have 1 generation per year. Collection sites can be scouted for wasp presence in mid to late summer by observing galls. Sites should have plenty of knapweed with a high proportion of galling. The collection site should not be sprayed with pesticides, grazed, mowed, or otherwise disturbed during the growing season. Make sure to scout several sites so that if a site is disturbed, you will still have a place to collect in the winter. The wasps are best collected by collecting stems containing galls in mid-winter. For collection, you will need handheld clippers in either large cardboard boxes, paper grocery bags, or another container that won't collect too much moisture. Gloves and long sleeves are recommended. Find stems that contain galls, and using the clippers, cut the stems near soil level. Place the stems in the boxes or bags. You may cut off any seed heads or side stems that do not contain galls. A couple of methods could be utilized for sorting releases, depending on your needs. If you are wanting to move the wasps only a few hundred meters on the same property with the same plant and insect community, you could just clip gall-containing stems and immediately set the galled stems in the new patch. This will allow the wasps inside the gall to emerge naturally based on temperature cues and is the least amount of work. This is an easy method, but there could be downsides. One is you will not know how large of a release of wasps you are introducing into the new patch. There could also be parasitoids within the galls. Gall-forming insects are often the target of parasitoids. Parasitoids are other insects that will lay their eggs either on or inside their host, killing them as the parasitoids feed and develop. To prevent the movement of parasitoids between sites that are separated by long distances, you should collect the galls and rear out the adult wasps indoors. This will allow you to kill the parasitoids as they emerge and then release only the adult wasps in known quantities. To rear out wasps indoors, you will need insect rearing cages, an aspirator, a spray bottle with plain tap water, a clicker counter, and release containers with either crumpled paper towels or some aspen fiber for nesting material. Insect rearing cages are mesh containers that allow for lots of air movement and light, but the mesh is fine enough not to allow the insects to move in or out. Begin by sorting the collected stems containing galls. Galls can be sorted by breaking or cutting off the lengths of stems that do not have galling and counting individual galls or weighing them. Don't break open the galls themselves, as they have developing larvae inside. The sorted galls can be kept in paper bags inside of a refrigerator around 40 degrees. Caution should be taken to make sure that the galls do not mold, so don't overstuff the bag or collect wet material. Periodically check in during storage. When the galls are brought out to room temperature, place them into the insect rearing cage. Try to time placing the galls at room temperature when the first emergence or sprouting of Russian knapweed at the intended release site occurs. Within about 2 weeks at room temperature, you will begin to see parasitoid emergence from the galls in the cage. We have several parasitoids in Colorado, but parasitoids have been observed in all western states where Aulacidea is established. All of these parasitoids are adventitious, meaning they occur naturally, and are generalists that attack just about any gall-forming insect. Parasitoids may move very quickly through a jumping motion and move their antennae quite fast while on the ground. Aulacidea are very slow and use their antennae to gently tap the ground. You can always use an aspirator to do a non-target or parasitoid collection in the cage of stems. Anything that is moving very fast or is definitely not Aulacidea should be suspected of being a parasitoid. These collected parasitoids can then be frozen or destroyed. Parasitoid emergence should lessen and stop as the adult Aulacidea begin emerging after about 3 weeks at room temperature. If the number of parasitoids has decreased enough, you could release the entire cage contents at your new site. This assumes the parasitoids are at a super low number and you are releasing in the same general area as the collection occurred. However, to completely avoid releasing or moving parasitoids, you might want to collect specifically only Aulacidea wasps daily from the cage. If you place a slightly damp paper towel within the cage, the Aulacidea will congregate and easily be grabbed from the paper towel using an aspirator. A spritz of plain water from a spray bottle can stop the wasps from moving or flying, making it much easier to count and collect them. We typically collect 200 adults and place them into a cardboard container with a small bed of nesting material. Try to collect wasps daily from the insect cages so the sorted containers can be placed in a refrigerator. Since the wasps only live around 5 days at room temperature, sorting and refrigerating the wasps daily is really important. This hand sorting prevents any chance of parasitoid release at a new site if you are careful about sorting. Almost all of the wasps will have emerged within the cage in a period of around 2 weeks, or 5 weeks after being brought to room temperature. When all wasps have emerged, the contents of the cage can be discarded. There should be no live parasitoids at this point in the galls, so if you want to place the remaining galls in the field at this time for late emerging Aulacidea, it should be okay. Releasing the sorted stem galling wasps is incredibly straightforward. Prepared releases in cups can be stored for up to 2 weeks in a refrigerator, but releasing as close to the collection time is better for the health of the insect. Remember, adults only live about 5 days, so the containers cannot be left at room temperature. Take your release cups into the patch of knapweed you would like to introduce insects. Usually mid-morning on a sunny day is the best time for release, as the insects will be more active, but any time should be okay. Don't release in very heavy rain, but if it rains after release, it is okay, the insects will be fine. Open up the container and tip the insects out into the knapweed all in one spot. They will disperse themselves throughout the patch, so you should not scatter them. If they are clinging inside of the cup, you can take a bit of grass or soft foliage and gently brush them out of the cup. Any nesting material can be left in the field, as it is biodegradable. We usually recommend a release of 200 stem galling wasps per 1/8 acre of infestation. I like to take a photo of the weed patch for future reference, a GPS point, the number and species of insect released, and the date of release for my records. Stem galls can be seen within only a couple of weeks after release or adult emergence in spring. The galls will continue to expand and harden throughout the summer. These galls act as energy sinks for the plant's resources, which can stress the plant. As the plant is under attack, it will try to send up as much new growth as possible, increasing the side shoots of the plant. These new growing tips are a perfect site for the Rosette Galling midges to lay their eggs and form secondary galls on the plants. It has been shown that in areas where gall wasps have been released, the population of gall midges can rapidly increase. This is such a great 1-2 punch for control of Russian knapweed. Flowering and seeding may be heavily impacted, which will help prevent spread of the Russian knapweed in the landscape. Over time, as the root resources are used for gall formation and not replenished because of diminished photosynthesis, the plant may become stunted. Both types of galls and multiples of each can be found in singular plants, which can heavily distort the growth and make Russian knapweed almost unrecognizable. These will slowly, over the course of 3 to 5 years, decrease both Russian knapweed abundance and competitive ability, allowing other control measures to be integrated. Eradication of knapweed from your land will take an integrated approach. Not all control methods will be compatible with the stem galling wasp or the Rosette Galling midge, and the biological controls themselves may not work in all areas. Mowing or plowing are not compatible with knapweed stem galling wasps, as it destroys the single generation of developing insects. However, mowing can increase gall midge populations, as it spurs the new growing shoot tips they need to lay their eggs. Herbicide may be compatible if applied in very late fall, typically after the first freeze. Coincidentally, this is typically the recommended timing for herbicide application. It's possible biological controls can reduce the Russian knapweed population to a point where other control methods will be more feasible or affordable. A healthy landscape with lots of competitive native vegetation may see better and faster results from biological control. Strategies to increase natives could include tillage-free reseeding and providing supplementary moisture during the growth stages of desirable plants. I hope this video has given you the information, methods, and confidence to identify, collect, release, and manage Russian knapweed stem galling wasps and Rosette Galling midges. There will be a list of references and sources in the video description if you would like to read more about these helpful insects and how you can manage knapweed. Thank you for joining me today.